This video will be on protein structure and on some historic experiments that looked at how proteins are encoded. So proteins are made up of amino acids. They're also called polypeptides, and all amino acids have the same basic structure. So they have an N followed by two C's. The N is called the amino group. It has two H's attached to it, and the carbon on the other end is a COOH. So it has a carboxyl group attached to it on every single amino acid. And the place where each amino acid differs from each other is in this side group, which is designated by the letter R here. So there are 20 different amino acids that are commonly used in proteins, and each of these amino acids has a three-letter abbreviation. Okay, so these are shown here. Also a one-letter abbreviation. And so when we look at protein sequences, which we'll be doing in lab later on in the course, what you'll see is this one-letter protein abbreviation or amino acid abbreviation. These amino acids can also be separated into different functional groups. So for example, this top group are hydrophobic side chains. Okay, middle ones are hydrophilic, and then there are also acidic side chains and basic side chains. When proteins are synthesized, the carboxyl group of the first amino acid is linked to the amino group of the next amino acid. This happens in ribosomes, and this formation of this bond releases a molecule of water, and a peptide bond is formed between that carbon and that nitrogen. So every single amino acid then is lined up NCC, NCC, etc. And so the result is that you end up with a protein that is polarized. So on one end is the amino end, or it's also called the N-terminus, and on the other end is the carboxyl end, or the carboxy terminus, or C-terminus. So once proteins are made, there are a number of different ways that they need to be formed into certain structures. And so there are different levels of structure, and I'll just talk about these four quickly here. The first level of structure is simply the order of amino acids. Okay, so you have a methionine followed by a glutamine, etc. Secondary structure involves some very simple structures such as an alpha helix or a pleated sheet. So these are shown here. And these are the most common forms of secondary structure. And what I just want to point out is that usually these types of structure generally do not use the side chains to make this alpha helix, for example. So we're just looking at interactions within those basic, NCC molecules of the amino acid. Tertiary structure, in contrast, does involve the side chains, and it involves things such as ionic interactions, plus minus interactions. Different side chains might have hydrophobic interactions, or make a little bond between two sulfurs. Okay, so things like that. And then finally, quaternary structure is when you have a protein that actually joins together with other proteins to make this multimer. And so, for example, hemoglobin is a molecule that you need four of these proteins in order to make a functioning hemoglobin molecule. Next, we'll talk about some historic experiments looking at how proteins are coded. So back in the 1960s, it was really not understood at all how we could go from a nucleotide code that's found in the DNA and get that translated into an amino acid code or a protein code. And so some of the early experiments that were really important we'll discuss here. So first, Yanovsky, 1963, he made some mutations in a gene in E. coli. And so these are the mutations that he made, right? He made these mutations in the gene, And what he found is that he was able to map each of these genes and the position of the mutation. And then he looked at the protein that was encoded for by that gene. And what he found is that those mutations occurred in the same order, right? So he had a mutation early on in the gene, and he found that, in fact, there was a mutation early on in the protein as well, near to the N-terminus of the protein. So this really proved that genes and proteins are collinear. Next, people were very interested in trying to figure out how is it that a simple code of only 4 bases can provide the information to call for 20 different amino acids. And so there were different ideas. So you guys all know now that it's a triplet code, so 3 bases code for 1 amino acid. But this was not known back in the 1960s. And so, some of the possibilities were as follows. So, if you took 2 nucleotides, problem is, that's not really enough possible combinations for all 20 amino acids. 3 nucleotides, on the other hand, seemed like, well, that's too many, because if you have 3 nucleotides calling for 1 amino acid, that actually has 64 possible combinations, and there's only 20 amino acids. Another possibility that people considered for a little while is that it could be an overlapping code. And so what I mean by overlapping code is illustrated here. So if you start out the beginning, so the first codon may be these 3 bases AUU, okay, that may code for an amino acid 1. And then if you just slide down 1, the next 3 bases are UUG, all right, and this might code for the next one. Okay, next one would be UGC, etc. This was quickly disproven. This was one of the first ones that people figured out that was not correct, because they found that when you had a mutation on the DNA level, that mutation would only affect 1 amino acid. So it couldn't be overlapping. Next, I want to talk about some important experiments that were done by Francis Crick and Sidney Brenner. And these were done in the 1960s, and eventually these experiments were able to prove that it is a triplet code. So what they did is they introduced mutations with a mutagen called proflavin into a gene in this bacteriophage. Proflavin either makes a deletion or an insertion of a single base, and what that leads to is a frame shift mutation. And so if you just look at this representation of, a triplet code, each of these codes spells a word, right? And we've got a sentence here that actually makes sense. And so if they would hit that gene with proflavin, you very frequently get a frame shift mutation. So what happened here is that you lost a C, and everything after that position is gibberish. Now, the important experiment that they did was they took these frame shift mutations and they hit them a second time with proflavin. And what they were looking for was something called a revertant. So a revertant is something that is a compensating mutation that fixes a frame shift mutation and puts it back into frame. And usually a revertant isn't necessarily a exact change back to what it was to begin with, but you can see here that so the C is still missing in that position, but there's a compensating A put a little bit later. Okay, so what that does that's so important here is that it puts the rest of the protein back into frame, and this protein, even though it has a few small mistakes still, we can make something that at least has some function. So what they found is that whenever your original mutation was a deletion, the revertant was always the addition of a base, an extra base, to get it back into frame. That's just shown here by looking at bases. So just to review this again, so you start out with wild type. This is the wild type sequence, and this is the protein that is coded for by that sequence. And if you do either an insertion or deletion, so in this case we're adding an extra base, then you get a frame shift. And you can get a compensating mutation, which is a deletion, all right, and now everything is back into frame again. You still have a couple of altered amino acids, but it's mostly, this protein is likely to have at least some function. So in class, we're going to talk a little bit more about this experiment and talk about exactly what this is proving, and also to talk about some further experiments that Crick and Brauner did in this same vein.